Hello and welcome to this feedback event. My name is Stuart Evans and I'm the Chief Examiner for the 9BNO or SNAB A-Level Biology. The purpose of this event is to provide feedback on candidate performance in the three 2023 examinations for this specification. I'll split the feedback into two parts. In the first part, I'll make some general comments on candidate performance across all three papers. Then, in the second part, I will consider some specific examples of candidate underperformance on the individual papers. This event should run for approximately 45 minutes. So we'll begin with some general comments. General observations about the examination papers are summarised on this slide. There was evidence of ramping in terms of demand. In general, candidates found the question demand increased within questions and through the papers. For all three papers, the mean and standard deviation increased compared to the pre-pandemic papers. Reasons for this include candidates having a better understanding of question taxonomy and stronger indirect practical skills. We've also worked on our mark schemes to make marking points more accessible, allowing candidates to better demonstrate their biological knowledge and understanding. Weaker candidates struggled to identify relevant information to include in their answers. Answers that gained marks often contained irrelevant information or incorrect information, as well as the required information to gain the mark. In contrast, stronger candidates tended to select and use appropriate biological knowledge and information. Weaker candidates, as opposed to stronger candidates, often demonstrated a poor grasp of basic or fundamental concepts. For instance, they confused terms such as transcription and translation, or gene and allele. Weaker candidates often did not attempt or made basic errors in calculations. There were, however, a subset of stronger candidates that also struggled with the calculation questions. More candidates appear to be providing workings, although in general these could be more clearly laid out. Weaker candidates often demonstrated poor written communication skills, although again there was a group of stronger candidates with poor communication skills. Many candidates used colloquialisms in place of scientific vocabulary. For example, they might describe sodium rushing into axons rather than something like sodium ions entering by facilitated diffusion. Some weaker candidates gave answers as a set of bullet points, and whilst demonstrating an element of understanding, these answers often lacked sufficient detail to gain marks. Many candidates gave a converse answer. For example, if they were asked to explain an increase in X, they provide an answer that explained a decrease, or they might just limit their explanation to a change. Weaker candidates also tended to ignore the context of a question and provided a generic answer. For example, they might explain the process of evolution, but fail to link their answer to the specific example used in the question. This can limit the award of marks. Having said all that, there is evidence that candidate performance is improving. And perhaps the main point, at least from our perspective, is that the examinations as a whole performed well. All three papers had reasonable mean marks and good standard deviations or spread of marks. This means that the papers allowed candidates to demonstrate their learning and allowed us to grade candidates fairly. Throughout this feedback, I'll be making reference to two groups of students, weaker or E-grade candidates and stronger or A-grade candidates. Now I want to look at some specific examples from each of the papers, beginning with 9BNO01 or Paper 1. Candidate performance in Paper 1 was generally better than in Paper 2 and Paper 3. This is probably due to candidates finding the content assessed in Paper 1 more accessible than that in Papers 2 and 3. The image on the right of the slide shows the facility or difficulty rating of each question part for E and A grade candidates, E grade being on the left and A grade on the right. Don't worry about individual items, but look at the general trends. Red means that as a cohort they found a question hard, and green that they found it easy. Essentially, candidates found the paper harder as they progressed through it, which is how it's designed, and also, again, not surprisingly, A grade candidates found the paper easier than E grade candidates did. We use the facility rating to help identify question parts that didn't perform as expected. The examples that follow have been identified using this approach. The first question part I want to look at is question 3A. This was a straightforward question asking candidates to demonstrate biological knowledge directly from the specification, an A01 type of question. A and E grade candidates found this question more demanding than we expected. 1 mark was targeted at E grade and 1 at grades between E and A. I'll use C as shorthand for between A and E. Three example responses are shown on this slide. In the top response, the candidate made reference to deoxyribose, base and phosphate group, marking point 1. The candidate has used the term inorganic phosphate, which is incorrect. Inorganic phosphate meaning free phosphate ions. However, as the question is targeted at E grade candidates, we ignored this error. Then, containing adenine, thymine, cytosine or guanine, together with deoxyribose, gained marking point 2. In the middle response, there's another error, this time a language error. Mononucleotide is made of adenine, guanine, thymine and cytosine. The candidate's implying that all 4 bases are present in a nucleotide. Again, the question's targeted at E grade candidates, so the error was ignored. In the bottom response, the candidate appears to have little idea about a very basic concept. In the specification, the arrangement of the different components is described. Very few candidates included this information in their answers. The closest we generally got was when candidates included a label diagram, as in response 1 at the top of the slide. The next example is question 4, part B. This was another A01 type question where candidates were asked to describe what happens inside a cell during prophase of mitosis. It was targeted at 1E, 2Cs and 1A. E grade candidates performed as expected and told us that the chromosomes were condensing. A grade candidates found the question more difficult than we anticipated, with many only giving the first marking point about condensation of chromosomes, and few A grade candidates could recall 4 different events that were taking place. These are fairly typical examples of responses from E grade candidates. The candidates appear to have a poor grasp of the biology. For example, in the first response, the candidate appears to think the centromere is part of the spindle or spindle fibers rather than part of the chromosome. In the second response, the candidate seems to think chromosome replication is taking place in prophase. This poor grasp of biological knowledge is a common theme in responses produced by E grade candidates. The lack of clarity of expression is also apparent in these responses. For example, the nuclear envelope starts to go, which leaves the chromosomes in the cell. Or the nucleus opens up. The next example I want to look at is 2B.2. Another question on paper 1 that many A grade candidates struggled with was the calculation of a diversity index. This question was targeted at grade C and above candidates. An equation for the diversity index is given in the specification. Candidates therefore could be asked to recall as well as use the equation. On this occasion, the equation was provided. However, even then, many candidates struggled to use the equation to find a value for D. Many candidates, even at the A grade, struggled to select the correct number for the numerator, and others did not realise that the value for the denominator was given to them in the table on the previous page, and wasted time calculating the value for themselves. In the top example, the candidate thought n represented the number of species rather than the total number of individuals of all species. In the second example, the candidates calculated their own denominator. It's correct, but obviously this additional step in the calculation takes time and introduces the possibility of errors creeping in. Now I want to look at some examples from paper 9BNO02 or paper 2. Overall, candidate performance in paper 2 is somewhere between that on paper 1 and paper 3. Let's consider some examples where candidate performance was different to that was expected. Again, these examples have been identified using the facility rating. We begin with two related question parts, question 1A.E3 and 1B. 1A.E3 was a multiple choice question and was targeted at E-grade candidates. Candidates were asked in which stage of cell division are chromatids first visible when using a light microscope. Both A and E-grade candidates found this question moderately difficult, and we're not sure why. Could it be because we included the phrase using a light microscope? We thought this would help avoid candidates worrying about chromosomes being visible using electron microscopes, but perhaps it acted as a confounder instead. Alternatively, perhaps candidates recalled the process as a sequence of steps with chromosomes condensing in the second step, so they went for distractor B, ignoring the terms. In question 1B, candidates were asked to explain how crossing over can lead to genetic variation. This is an A01 question and was targeted at 2 E grade marks and a C grade mark. Again, both A and E grade candidates struggled and found the question difficult. This response illustrates a few common mistakes that candidates made. Candidates often use the term gene when they should be talking about alleles. They describe the swapping as taking place between sister chromatids rather than non-sister chromatids, and they also seem to think that crossing over results in the reordering of genes. This response suggests a candidate that's familiar with the concept of crossing over, but is struggling to recall the biological ideas or detail needed to answer the question. And this is a common theme for many describe and explain questions that was seen across all three papers. Again, in this response, the candidate seems to have a recollection of the process, but can't provide sufficient detail to gain any marks. As in the previous response, the candidate uses the term gene in place of alleles. This response also illustrates how E-grade candidates often include a lot of irrelevant and incorrect information in their answers. This is an example of a better response from a stronger candidate that gained 2 marks. Stronger candidates managed to tell us that genetic materials swap between non-sister chromatids. They did not tell us that the non-sister chromatids came from homologous chromosomes, nor that the end result was a different combination of alleles. Okay, let's have a look at question 2b. 2b.1 asks candidates to describe what is meant by the term operon. 2b.2 asks them to describe the effect of lactose on the lac operon. Many candidates did not produce answers to these questions, or the answers they did produce lacked any relevant content. This may be because this is a part of the specification that has been assessed infrequently in the past. Candidates sensibly use past papers to help with their revision. However, they do need to be aware that questions can be asked that require recollection of biological knowledge from any part of the specification, and that because a part of the specification has not been assessed previously, this does not mean it will not be assessed in future papers. The next question I want to consider in paper 2 is question 3a. Question 3a was another multiple choice question that candidates found more difficult than anticipated. Candidates were asked which of the following describe the structure and function of a ligament. Both A and D grade candidates struggled to identify the correct answer as ligaments being elastic and attaching bone to bone. The next question I want to consider is question 7b3. At the start of part B, candidates were given some context for the rest of the question. They were told that there was a recessive genetic condition of the eye and that this was due to a non-functional gene. They were also told that genetically modified viruses can be used to treat people with this condition. In B part 3, candidates were asked to explain why a person successfully treated using a genetically modified virus may still have a child with this recessive condition. So this required them to use the context or provide an answer in the context of the information provided earlier in the question. This was an AO2, an application of understanding, and was targeted at 2C and 1A. Many candidates produced responses that lacked sufficient detail. Fairly typical were statements about the parent, in which candidates failed to identify if they were talking about the person treated or their partner, and many made reference to genetic code but did not distinguish between the eye and gametes. Few candidates in fact made any reference to gametes at all. Candidates at the E grade often ignored the context of the question and simply told us how a recessive trait can be inherited. A grade candidates generally told us that only cells in the eye were being genetically modified. Very few went much further, and they did not consider gametes or carrier status of the other parent. Now I want to consider one of the levels-based questions on paper 2, question 10b.2. There can be up to 2 6-mark level-based questions on papers 1 and 2 and 1 9-mark level-based question on paper 3. 10b.2 is a fairly typical level-based question in which candidates are provided with some information and then asked to use the information to help produce an answer. The question they were asked was to evaluate the information provided in the table and graphs to determine which factors are most likely to increase the risk of CHD in women. The type of response a candidate needs to provide depends on the command word used. In general, candidates are getting better at matching their answers to the command word used in the question. This question asks candidates to evaluate the information provided. Candidates will be assessed on their ability to bring together information to reach a judgment or conclusion. Candidate responses are assessed using the level-based mark scheme. This is the main part of the mark scheme and as you can see, the quality of the conclusion reached and the candidate's use of information provided to support that conclusion are what determines the level and therefore the mark awarded. Candidate responses are first judged against criteria to determine the level of response. Then the quality of the response is assessed to determine the mark to be awarded, e.g. level 2, 3 or 4 marks. The second paragraph in each level box describe the criteria required for that level. In the Evaluate mark scheme, a level 3 response requires a conclusion and should demonstrate sustained linkage to biological knowledge and understanding. Mark schemes also contain a section entitled Indicative Content. As the title suggests, this is simply an indication of the points candidates might make. Indicative content should not be seen from the perspective of points-based mark schemes. Candidates could make many of the indicative content points but still be restricted to a level 1 or level 2 response. In some mark schemes, for level-based questions, there may be additional guidance. This additional guidance gives an indication of how the level of a candidate response might be determined. However, this is only guidance and it does not replace the main mark scheme. Candidates were provided with some information in the form of a table describing age, BMI and the incidence of CHD in women. This was in part 1 of the question, .E1. Not all candidates referred to this data when they were answering 10b.E2, even though they were prompted to do so in the question. Candidates were also given 3 graphs describing the effects of BMI on the incidence of CHD for women that smoked, were inactive or drank alcohol. They were then asked to evaluate this information. We'll start by looking at a level 3 response. The first part of the student response is shown on this slide. This is the second part of the candidate's response. In this response, the candidate has made relevant statement based on all the information provided in the table and in the graphs. They've produced a general conclusion supported by these statements. This becomes a level 3 response. The question then is there a reason to limit the response to 5 marks? The relationship between BMI and smoking on CHD could have been described in more detail, and the use of the term significant difference when no error bars are provided keep this response at 5 marks. This example was considered to be a level 1 response. The candidate has only made basic comments using 2 lifestyle factors, smoking and activity. They've not considered age or BMI and have not made any attempt at an overall conclusion, limiting the response to level 1. The decision then becomes, is there a reason not to award 2 marks? As the candidates considered more than 1 lifestyle factor, they were awarded 2 marks. Now I want to look at some examples from 9BN03 or paper 3. In terms of raw marks, candidate performance on paper 3 was generally below that on paper 1 and paper 2. This is not unexpected, as questions on paper 3 tend to be more synoptic, have a greater emphasis on practical elements of the course, and include questions on the pre-release article. So let's consider some examples where candidate performance was different to that was expected. Again, we've used the facility demand to help us identify some questions to look at. To start with, we're going to look at two parts of question 1, 1B.E2 and 1B.E3. Both proved more difficult than anticipated for candidates at both the A and the E grade boundaries. Okay, so this question, candidates had been given some context previously, and they were now asked to explain why the left atrium stops beating when it's separated from the right atrium, and .E3, explain why the right atrium and the rest of the heart will eventually stop beating. So these are both AO2, so application of biological knowledge and understanding, and they're targeted at 1E and 1C grade mark. E grade candidates found these questions to be more challenging than we anticipated. A grade candidates found them relatively straightforward. This slide has 3 responses that are fairly typical of E grade candidates. Many candidates seem to think the heart beats because its chambers fill with blood, and candidates often ignored the role of the SAN in coordinating contraction and its location in the right atrium. Candidates above the E grade boundary found this question to be relatively straightforward and approachable, and this is a typical response of a candidate at the C or above grade, where they've clearly identified the role of the SAN and its location and understand this in the context of answering the question. Okay, now I want to have a look at question 4C. So this question is an AO1 style question where candidates are asked to describe how the production of sweat is controlled during exercise in humans. A-grade candidates found this question more difficult than we anticipated. It was targeted at 2 E's, 1 C and 1 A-grade mark. This is a fairly typical example of a response by a candidate who clearly knows the story but is not providing the required level of detail. They've not told us where the thermoreceptors are located, and they haven't told us the result of stimulating sweat glands is an increased rate of sweating or increased sweat production. The candidate has also included information about shunt vessels, arterioles and blood flow to the surface of the skin, information not required to answer the question. And this is a better response where the candidates gained marking point 2, 3 and 5, so 3 out of 4 marks. Now I want to have a look at question 5A.E3. This question was one that A-grade candidates found more difficult than we anticipated. It's a relatively straightforward AO1 style question. Was targeted at 2 E-grade marks, 2 C-grade and 1 A-grade mark. And the question was describe the role of ion channels in the conduction of a nerve impulse. The majority of candidates, like this one, clearly have an idea of what's happening. However, their answers lack the required detail and sometimes suggest the candidates might hold significant misconceptions, particularly about what's being depolarized or repolarized. and the mechanism by which the ions are moving across membranes. Many candidates fail to describe ions moving by diffusion. Instead, they often use terms such as ions rushing in, or, as in this case, ions flooding in. A surprising number of candidates described ions being pumped in or out of the axon. Candidates frequently described ions moving into and out of the membrane rather than across or through the membrane, and many candidates gave answers in terms of sodium or potassium rather than the metal ions. Candidates also frequently made statements about depolarization or repolarization without locating these events to the membrane, some even suggesting that the whole cell is being depolarized or repolarized. This is an example of a better response that included the required detail. Now I want to have a look at question 8F. 8F is one of the questions based on the pre-release scientific article. This question asked candidates to explain how chloroplasts are adapted to their role in photosynthesis. The question was targeted at 1E, 2C and 1A grade mark. A grade candidates found this question much more difficult than we anticipated. The majority of candidates ignored the command word explain and instead provided descriptions of chloroplasts. The mark scheme was adjusted before marking began to take account of candidate responses. Even then, many candidates struggled to gain marks. It's important to realise that if a similar question was asked again in a future paper, the mark scheme would not necessarily be as generous. Some flexibility in the requirements of mark schemes was introduced to make allowance for recent disruption to learning caused by school closures during the pandemic. Candidates should be aware that it's important that explain questions are answered with responses containing reasons and not just descriptions. This response scored well. Although we would have liked to have seen more by way of explanation, for example, of the importance of the large thylakoid surface area allowing for the maximum absorbance of light, or the proximity of stroma surrounding the grana allowing for rapid transfer of the products of light-dependent reaction to the enzymes involved in the light-independent reaction. That concludes my feedback on specific examples of questions that candidates found more challenging than we anticipated. To finish this feedback session, I now want to point out opportunities we see for candidates to improve their performance in the examinations. Well, what general conclusions can we draw from this examination series? First, it was pleasing to see that candidates tended to perform reasonably well on data interpretation questions. And secondly, candidates performed much better on questions assessing indirect practical skills compared to the 2019 pre-pandemic cohort. However, many responses lacked sufficient detail or contained irrelevant detail. Candidates often used biological terms incorrectly. Many candidates provided a describe answer to an explain question, and many candidates seemed to hold significant misconceptions. What does this mean for candidates in future exam series? Candidates can improve their marks if they read questions carefully and make sure they answer the question asked, and that's particularly important in terms of command words. So an explanation or a description require different types of answer. They need to make sure their answers contain sufficient detail, and they need to ensure they understand and use biological terms correctly. Thank you for listening to this feedback session. We hope that it will be useful to teachers and students following the 9BNO A-level specification.